kick it off straight away. I have the pleasure, here he is. I have the pleasure to introduce Ryan Carvalho. He's actually, thank you so much also for providing us here as a host, research facilities. And Ryan is the head of Nestlé Research, and he is a trained pediatric gastroenterologist by training. And yeah, Ryan, over to you to introduce our next session.
So good afternoon, everyone, and welcome back. We're hoping to have an engaging and productive session this afternoon. This morning, we started with the topic of meeting nutrient needs in a changing environment. And we heard from multiple speakers that what we eat is food and not nutrients, that have impacts on our health, nutrition, but also the social and psychological benefits of food. In this session, we're going to start by talking a little bit more about the role of meals, from their role in nutrition to the social aspects. We'll have three esteemed speakers. The first will be Dr. Kate Schneider, who will be presenting virtually from Johns Hopkins University in Baltimore in the US. She will speak about meeting nutrient requirements when meals are shared, the importance of affordable nutrient density of foods. And this is a critical topic for all of us, because nutrient requirements are set for individual needs. But across the world, most families eat shared meals. So how do you adapt the nutrition requirements and nutrient requirements to meet the shared meal occasion? The second speaker will be Professor Ciarán Forde, who will speak about how sensory cues influence our eating behaviors, food intake, and health. And then we cannot end the session with talking about how should we look at meeting nutrient requirements, the nutrition and social aspects in the world where we live in close compatibility with AI. And to answer those questions, we have Dr. Kosmas Dimitropoulos, who will speak about how we can use AI to optimize meal plans to meet dietary needs. With that introduction to the session, let me introduce our first speaker. Dr. Kate Schneider is a research scholar at the School of Advanced International Studies at the Johns Hopkins University. She currently leads the data team for the Food Systems Countdown Initiative, which is a large collaborative effort of over 60 multidisciplinary scientists from many institutions working to monitor and understand food systems and their transformation to meet the 2030 global goals. She also collaborates with the UN Food and Agriculture Organization and other scholars on multiple projects related to understanding employment and value-added work in agri-food systems. Today, she will focus a lot on how her work on understanding the role of prices and preferences in diet quality outcomes, mechanisms that protect access to nutrition, and data gaps and how we can creatively solve them. With that, I'll hand it over to Dr. Kate Schneider. Thank you for your time this morning, this afternoon, morning for you in the US, and we look forward to an insightful presentation. Thank you.
Thank you so much for that introduction. Are you able to hear me?
Yes.
Yes, okay, great. Let me just share my slides here. All right. Thank you. So I'm Dr. Kate Schneider. I'm a research faculty member at Johns Hopkins and joining you from Washington, D.C., where I have surprisingly poor internet connection. So I will just apologize in advance. If my internet drops, I'll be right back. And so sorry for the disruption. I'm pleased today to present some work that was actually from my dissertation. So I'll be honest, it's a couple years old here, but I think still very relevant on meeting nutrient requirements when families eat shared meals. So this work was motivated by some specific context in Malawi itself. When I started this work, there was a brand new multi-sectoral nutrition policy and strategy, which articulated a number of information needs for the country to design and implement and monitor plan programs to meet its nutrition goals. There had been substantial progress on stunting in Malawi that over the last decade, but it had been noted that a lot of the gains were slowing. And so this was kind of the broader context motivating the multi-sectoral strategy that they put in place. And so it was a moment when the government showed political will to address malnutrition. So that was sort of the Malawi-specific context. And these are the information needs that were articulated in that document. And we hoped that this work could help address some of these here. There was also a broader motivation and kind of broader motivations about, what methodology could be brought to bear on this type of question that could be really broadly applicable to other places as well. So we have a number of existing data sources of the household survey type. So these can provide an information source that could be further explored to answer nutrition questions, but hasn't really been fully exploited to date to do so. We also have least cost diets, which are the lowest cost diet that meet a set of nutritional constraints. And so in our case, we're considering nutrient adequacy. So I totally agree with everything you heard this morning. People eat food and not nutrients. Despite that, we did this study at the nutrient level to try and get a sense of what is really the difference between when you're planning for somebody eating alone and a group of people eating together who have very different nutrient requirements. So these least cost diets, this is not a new method, but they've mostly been done for a single person. What's the lowest cost diet to meet the nutrient requirements of this individual, or the food balance across food groups to meet recommended dietary recommendations. And the group that I was working with at the time had done this for representative women around the world, but it hadn't been done for all people of all different age, sex, and physical activity groups, and it certainly hadn't been done for the context of people who are eating a shared meal together and who have different needs. Sorry, one more point there. So we combined household survey data with this least cost diet approach to extend the least cost diets from individuals to households. We had 3 research questions motivating this study. The first one is, what's the level of diet quality that would meet the needs of all members? So how nutrient dense would their diet have to be, and how does that compare to current diet? So we developed this shared diet benchmark, a set of nutrient requirements that we could then apply to the household. And then in the second question, we wanted to ask whether or not these diets are available and affordable year-round. We did this in the context, as I mentioned before, of rural Malawi, but the methods here are broadly applicable. And then in the third, we asked what are the drivers of what we found, which is that the shared diet is infeasible and of quite high cost. And so we looked at what policy solutions are available. So to answer this first question, we developed that shared diet benchmark, and we looked at how that benchmark compared to current diets. I'm not going to focus as much on those results because I wanted to get to the more interesting ones, what I think are the more interesting ones about, why is a diet that meets these requirements not possible right now, and actually what could help improve the situation. And so the results that I'm going to focus on here ask what an adequate diet costs relative to household incomes and how much it varies seasonally, and then what drives those results and policy options that could actually help improve the situation. So just a bit on the data here. We merged several data sources together. So we start with these nationally representative household survey panel data, and we combine those with monthly retail market food prices that we have for 27 markets around Malawi. We then used newly compiled food composition data that was specific to Malawi. And then we used the scientific nutrient requirements that I'm sure you're all familiar with, defined in the dietary reference intakes. So how did we come up with this single requirement for a combination of individuals in the household? So we developed these household nutrient requirements based on a diet that has the nutrient density to satisfy the neediest member. So we start with the individual requirements for nutrients, which of course, as you know, vary by age, sex, maternity status, and physical activity. And then we take for every household, we look at its actual observed composition, and we create a diet that works for that specific household. So we did this for the households that we observed in the survey, not some sort of generic household composition. So we had a variety of households. So we define the diet that each household needs on the basis of nutrient density. So the level of nutrient density in the shared household diet is set at that required by whichever member has the most demanding need for things with a lower bound, or the most restrictive upper tolerance for things with an upper bound for nutrient density. And we define that per nutrient. And then the energy need is the sum of all energy needs for the household. So for those who like to see this in math, economists like to show our math. So here you go. Here's how we do that. Establish that household requirement. And what this leads to is a diet with sufficient total energy for all of the individuals, where if everybody eats a share of the diet from the household diet that meets their energy needs, all their other nutrient needs will be satisfied and will not exceed any of their upper bounds. Now, I'm not going to present the results of how this compares to current diets, so I'll just tell you that when we looked at what families were actually eating in rural Malawi, this diet would have a lot more riboflavin, selenium, lipids, and B12. It would have a lot fewer of its calories from carbohydrates, and it would have less copper. So what that's telling us is the diets are lacking in 4 nutrients, have too many carbohydrates, and have too much copper. Turning to the cost of that diet, we then apply the nutrient requirements as the constraints for a least cost diet optimization problem. And so this is a linear optimization problem where we say, what's the lowest cost? We give it all the foods at whatever prices that have been observed in the market. What's the lowest total cost to get a diet that meets those shared nutrient requirements? And we estimate this as, again, here's the math if you like to see it. But basically, it's just select from all of these foods and their quantities a basket that will meet the nutrient requirements that we set. And we do this for every for every individual meeting their individual requirements. And we do it for every household meeting those shared nutrient requirements based on the composition that was observed in the survey for every month from 2013 to 2016. And so when we solve the optimization problem that you see here, what we get is a combination of foods in specific quantities that meet the set nutrient requirements. So they stay within the lower bounds and the upper bounds, and they meet everyone's energy needs at the lowest total cost for each month and for every household in the market that's nearest to where they live. The district central market is how we define that. So the first question we wanted to answer is, are the nutrient adequate diets affordable year round? And the way we answered this question, and this is the last bit of methods here, is we thought about this from a bounding approach. So the lowest cost way for a household to meet all members' needs would be for every person to eat a separate meal that was optimized to their individual needs. And so we could estimate the cost of the individual diets in the household and add that up. And that gives us a lower bound on what's the lowest cost at which this household could meet everybody's nutrient requirements. And so we call this the individualized diet scenario. And then we estimate the cost of the shared diet that I just described as the upper bound on the cost, because in this context, some members of the household will actually get more of the nutrients than meeting their minimum needs if somebody else is needier in terms of nutrient density. But we've made sure that nobody's going to exceed their upper bound tolerance. And so this is the shared diet scenario, but it's going to cost more because there's more nutrient density in this diet. And so this gives us the metric against which we can compare household incomes. And so affordability is the estimated cost of the diet relative to monthly food and total spending, which we have observed in the household survey. And so this can give us a range to assess affordability. We also argue that thinking about this as a range between the individualized cost and the shared diet cost, it's more realistic than if we were to look at either bound alone. And then we also have data that on these monthly price fluctuations, so we can understand how much both of these diets, both bounds, vary seasonally. All right, turning to our results here. First, we look at feasibility. So can you even identify a basket of foods that meet these requirements from the prices and items observed in the market? So what we find for those shared diets is that they're only feasible in the market 60% of the time. So the feasibility of the household shared diet by month and scenarios in those dark lines And that indicates the percentage of the households with a feasible diet in the month under the, the light blue lines is the shared scenario and the dark blue lines is the individualized scenario. And this is population statistics, which are corrected with sampling weight. So this is a picture on average for all rural households in Malawi. How feasible is
Sorry, I think it's frozen here.
It's her. She said that her line is back and get back.
much lower feasibility, and what you see in the dashed line is the average for the individualized diet's feasibility, and the solid line is for the shared diet's feasibility. And we defined a diet as feasible under the individualized scenario only if everybody in the household had a solution to the individualized diet. So that's what makes the individualized diet infeasible most often, is there's at least one member of the household for whom their nutrient requirements could not be met. And that was more often, of course, I think of no surprise to this audience, people who have the greatest nutrient requirements. So young children, children under 5, toddlers, and pregnant and lactating women are most often the ones whose nutrient requirements have the hardest time being met, even for the individualized diet. Okay, so what does it cost? So as you, I think, would be expected, the individualized diet is lower cost. It has a median cost of $1.75 per person per day, compared to a median cost of $2.26 per person per day for the shared diet. We do see evidence here of a seasonal pattern. We formally tested that. I'm not going to show you those results in the interest of time. But what we did find was that seasonality in the cost of the shared diet was about 11 times greater than the individualized diets. And the greatest seasonal gaps, so the food items that have the greatest fluctuation over the course of the year in their prices, are fruits and vegetables. All right. The affordability part, this is the most interesting piece of the results to me. So what we're looking at here is a fraction of households who cannot purchase the adequate diet in the month of their survey, either because it costs more than their total income or because it's not available. So the optimization problem could not arrive at a solution given the foods that were observed in the price data set. And we're looking here at the affordability under each scenario. And we're only looking here at the month that the household was surveyed, when we observed their total and food spending. So if the diet was infeasible, so the linear programming could not arrive at a solution, then we imagine that's like it were to have infinite cost. And so it's therefore unaffordable. So we count infeasible and unaffordable essentially as a similar outcome here. If it were feasible, then we compare the cost to the household's reported food and total spending. And just for context here, the households in our study are already spending 74% of their resources on food for the median household. So what we find here is that 44% of households cannot afford the lower bound diet cost, even if spending all of their resources on food. So these lighter bars are comparing the cost of the diet to their total spending. The darker bars are comparing the cost of the diet to their current observed food expenditure. So then there's another 18% for whom the lower bound diet is unaffordable without increasing their current food expenditure. So then in total, we have 62% of households in Malawi who couldn't afford an adequate diet at all without spending more on food. So this is the individualized diet. This would be the very onerous way of optimizing your resources. You give everybody a separate diet that perfectly met just their minimum needs, and there's still 62% of households that can't do this. We can also look at the other extreme. There are 20% of households who find the shared diet affordable within their current food budgets. So that's great. That's 1/5 of the population, but what about the other 80%? The other 80% can't afford the shared diet either because it's infeasible in the market or because it costs more than their current food spending. And for 69.5%, sorry, I got behind myself here, 69.5% for whom it costs more than all available resources. So that's the top of this dotted red bar that you see here. So that leaves the group of households in between these two dashed lines. That's 10.5% of households who could possibly afford the shared diet, but only if they spend more on food than they currently do. Those are those who's in this bar, people who can't afford the lower bound diet without increasing their food spending, and those who, it's between those who can't afford, the lower bound diet without increasing food spending, and those who can't afford the upper bound diet without increasing food spending. So this is 18% of the population who could afford to meet the family's nutrient needs within current food spending if they were really carefully allocating their resources to achieve that goal. But in doing so, they wouldn't be fully sharing their meals in accordance with social norms. So I'm sure you're asking, well, why? What is driving this cost, lack of feasibility, and the variation across different households? And so the first hypothesis that we looked at here was household composition. And so we wanted to understand whether the different types apologies, that was my terrible internet connection. So we looked at the different types of members living in households together. And so we call that household complexity. So how many different types of age and sex groups do we have in a combination in the household? And then we looked at how that's related to overall household size. So this figure on the left here shows that household membership complexity increases with household size, as one might expect, but only up to about 10 members, after which additional members are less likely to change the nutrient density requirements of the household. So in other words, household size and household complexity of membership are interrelated. So larger households aren't just scaled up smaller households, but they're likely to require greater nutrient density because they also have more complex membership. And on the right, we see that as households grow in size, which as we've just shown is also likely in complexity, the cost of 1,000 calories of the adequate shared diet increases. So we have one understanding here. What's driving this variation across households is it's a lot more expensive for households that have more people because that increases the variety of nutrient needs in the household. And that, and by doing so, increases the nutrient density requirements, most likely. The more members they have, the more likely they are to have those members who have the greatest nutrient density needs, small children, and pregnant and lactating women. We also wanted to know what were the nutrients behind driving these costs, so that this could give us some sense of whether or not there's a fortification strategy that could help reduce this cost. And so what we did here is we used what are called the dual results of the optimization model, and that can tell us the shadow price of each constraint. So the shadow price is kind of what is the percent that the diet cost would change for a very small change on the margin of this nutrient requirement, either up or down. And so what we find here is that the cost rises $2.50 per household per day for a 1% increase in riboflavin needs. So that is by far the most expensive nutrient that we observe in Malawi's food system. Whoops, sorry, I thought I had some animation there. Second to riboflavin, we see B12 also driving that cost, but the order of magnitude is so much lower here. It's 14 cents increase in the total diet cost per household per day on average for a 1% increase in vitamin B12 needs. And then at the upper bound, we also see if you let 1% more copper in the diet, so copper enters our equation in the upper bound constraint. So as if you remember, a lot of the households we observed in their current diets were actually consuming more copper than they should relative to that shared diet benchmark, but even actually relative to individualized diets as well. And we can see that here coming through in the cost as well, that if you let 1% more copper in the diet, it lowers the diet cost by 24 cents on average. So what is that telling us? It tells us that the diet gets more expensive by trying to avoid exceeding the copper upper tolerance for households. Okay, lastly, we wanted to look at some policy scenarios to better understand what could bring down these costs. We selected these scenarios based on the nutrients that were most underconsumed in current diets and costliest. So you can see the policy scenarios here in the box on the right. We basically just we messed with the price or availability of nutrient-dense foods. Eggs, dried fish, we observed only available in certain markets and months, and so we imagined by imputing a price into the price data set if it were available in all markets and all months. And then a similar idea with groundnuts, and also we lowered the price, and then we lowered the price of fresh milk, increased the availability of dry milk, and then we tried this last scenario here, selenium soil biofortification for maize, because it was currently under discussion by the government, and was something that was being actively studied by other researchers and carefully considered by the government. So what we found here is that most of these scenarios would have very little impact on the diet cost. And so what you're seeing in the figure on the left there is just the change in diet cost relative to the base case, which is as currently observed in prices and availability. And so we hoped that we would see a lot more bars kind of dropping the cost down, but most of them have very little impact. But the last one that we see here is that soil biofortification with selenium on maize fields would result in a near universal availability and half the average cost of the diet, and also nearly eliminates the shadow price of copper and slightly lowers the cost of vitamin B12. So what is this telling us? In order to meet selenium needs, the model draws on a lot of foods that are higher in cost to get enough selenium, but then it's also trying to work around not getting too much copper. And so when selenium needs can be met through the cheapest source of calories, which in Malawi is almost always maize, the model gets the other needed nutrients from their actual lowest cost options, not the options that would also add selenium and avoid adding more copper. So another way of thinking about this is that the shadow price of copper in the base case tells us that the model had to go to more expensive foods to get other nutrients in order to avoid getting too much copper. So we see selenium biofortification as a very promising policy option. As a very promising option. Whoops, I did have animation here. Apologies. All right, so I'm just getting to my last couple of slides here, and I hope I'll have plenty of time to take a couple of questions. There's a number of policy implications that emerge from this work, some of which are methodological and generalizable to broader contexts, and some of which are very specific to the Malawi case. So we see sort of two methodological contributions here, is that we've shown how to extend the least cost diets framework from individuals to households. And then we demonstrate that these widely available nationally representative household survey data on household composition and food composition can be linked with frequently collected subnational monthly food prices and local food composition data to estimate this bounded range of the cost of nutrient adequate diets that meet the needs for all members of a household. And then, of course, we find in Malawi specifically that these nutrient adequate diets are currently out of reach for most households, even if they were to go to we understand that this is explained in part by household composition and size, but it's also explained by some features of the food system. So we find riboflavin and B12 are the costliest nutrients, and good sources of those nutrients are dairy products and also eggs. But the selenium shortage is what makes the nutrient adequate diets infeasible. And some might argue selenium is only needed in this would potentially be another case that could be a good option for fortification strategy. But there's also this opportunity to engage in soil biofortification of maize with selenium, which would offer a promising policy option given that most Malawians are not getting their maize through any centralized system where you can implement a fortification strategy. And so biofortification at the field level would then introduce those nutrients into the crops that are then subsequently consumed by the household that largely by the household that grew the maize, or with local trading, but without ever passing through a centralized point that makes fortification a feasible strategy. Just a couple of concluding thoughts about why this matters. This is a very specific study in the Malawi context, but I think just kind of zooming out, like, what does this help us think more broadly about addressing complex nutrient requirements and nutrition challenges of our time? So we really need everybody to act more urgently, policymakers, citizens, and the private sector, in order to get access to affordable, healthy diets for everybody. We can't do this thinking with disciplinary blinders on. It really takes some creativity across fields, and also just thinking about how can we do the best with data that we have now, because a lot of the data that we have on what people eat, diet quality, nutrient composition, it's highly imperfect, but how can we do the best we can to make some progress on these urgent tasks, given that we do have a lot of data resources available to us if we make creative use of them. We always need more and better data, but that's just not an excuse to delay evidence-based recommendations for action, because the task at hand is too large and the challenge is so urgent to get healthy diets and better health outcomes for people around the world. So with that, I would like to say thank you, and I'd be very happy to take any questions.
So thank you, Kate. I think we're going to keep questions for at the end of the session after the next two speakers.
Sure.
So stay online. And I'm sure there are a lot of questions after that excellent talk, particularly I'm sure Adam's busting to ask questions about the math, particularly around shadow price computation. So we'll get there. Hold the questions till then. It is a pleasure now to move to our next speaker. Professor Ciarán Forde is the chair of Sensory Science and Eating Behavior Group in the Division of Human Nutrition and Health at Wageningen University, and the Research Division for Human Nutrition and Health in the Netherlands. He leads research on how food's sensory properties influence eating behaviors, energy intake, and metabolism across all life stages. He is currently the executive editor of the Journal of Appetite and section editor for the European Journal of Nutrition. He also holds editorial board positions at Nutrition Bulletin, Journal of Future Food, and Journal of Texture Studies. Today, he's going to share with us his research, which highlights the impact of odors, textures in stimulating sensory-specific appetites and behaviors. With that, we look forward to your presentation. Please welcome Dr. Ciarán Forde.
Thank you, Ryan, for the kind introduction, and indeed thank you, Petra, for the opportunity to come back to Nestlé and to meet my old friends and to talk about sensory, which is an uncommon topic in this program, and I think it's possibly, well, I believe it's the most important topic that we should be talking about. Believe it or not, today, somewhere in the world, there are nutrition conferences underway, and they're not talking about taste, and they're not talking about texture, and they're not talking about smell or choice or behavior. They're talking about nutrients in a vacuum. And as we've heard many times already today, nutrients don't exist, actually. Foods exist. And in fact, I'm pleased to be able to pull out a citation from my very first paper, the opening line of which I highlight that food has no nutritional value until it's chosen, accepted, and consumed, and indeed consumed over time. If we think about dietary patterns, and if we think about the way we learn food, we learn it through the sensory experiences of eating it and the relationship between that experience and the post-ingestive experience of fullness and satisfaction. And I'm reminded of this again and again as a researcher in the nutrition department, because when I speak to my nutrition colleagues, we talk about the nutritive qualities of diets. We talk about energy from micro and macronutrients. We talk about strategies to overcome excessive consumption. And there we have things like fortification or reformulation, much of which we've heard about today. If I talk to my public health colleagues, they'll say, well, we need to regulate the food environment. We need to think about portion size or calorie labeling, or maybe taxing unhealthy foods or stimulating the consumption of healthier foods. So we've identified problems and we've identified potential strategies to overcome those problems. However, today, we don't have any policies linked to how we interact with food. We don't have any strategic goals around how we eat and how we can account for differences between people. and how we could accommodate those differences in healthier diets that can actually promote the acceptance of more sustainable diets or even better nutrient intakes. And so how do we move from sensory, which is traditionally studied in a vacuum of just perception in isolation, how do we move from perception to ingestion and to think about how it could be used to shape healthier diets, healthier behaviors? Within this, I think of sensory as a non-nutritive influence of food intake, and I'll show you some data today that shows the profound and consistent impact of sensory experiences and how they shape how we pick our foods, how we eat our foods, and over time, how they shape our energy balance and our healthy or unhealthy diets. So we had earlier on from Professor Johnson the example of the John Blundell satiety cascade. I think this is really a guiding principle for many of us in the field of appetite regulation and food research. And here you can see that the sensory experience of eating actually occurs very early in a meal. It influences many of the events around how food is selected, how we select our portions, and even informs how we eat. Odors and visual cues direct us to food sources. Textures and tastes give us a clue about the freshness of a food or its nutritive density. Texture literally tells you how to eat the meal you're eating. It gives you cues as to how to adapt your behaviors. And all these events occur long before we have any serious post-ingestive or post-absorptive effects, and so we can really start to shape how meals are consumed using this kind of an understanding. Yet, as I mentioned, in nutrition today, very few conferences or discussions will be around the sensory properties of foods. And so I really think it's important to bring this back to this audience and think about how food choice and food intake behavior is largely shaped by the sensory experience and the cognitive aspects of eating, much more so than the nutritive properties of the food we're consuming. Yet at the same time, we can all agree chronic ill health and disease is strongly influenced by the nutritive properties of food. So diets with poor nutritive density, high energy density will lead to chronic ill health and many other examples that we've heard this morning around deficiencies. So my group's goal is to square that circle. How do we unite an understanding of how people perceive their food, how they make food choice decisions, how they eat their meals? How do we unite that with the nutritive properties of meals to enhance overall human health? So in the limited time I have available today, I'm going to talk a little bit about some quick examples from my own group in terms of the work we do on odours and taste. My own area is in texture, so I'll dominate the talk by lots of talk around how texture can be used to manipulate behaviour. And at the end, I'll give some examples of how I think we can translate this into actual policies or even attempts to redefine products to make them healthier in the future. So we'll give some quick examples from my own group on smell and taste, and then we'll delve into texture. I've also asked to cover a little bit about some of the ongoing work we have around how the eater interacts with the food, but also how the food matrix interacts with the eater to shape our metabolic response to the foods we consume. And I'll give some examples of that from our own work, and then offer some hope at the end about how we can use sensory cues to change eating behaviours. Because we can reformulate food, we can take out public health sensitive nutrients of concern. We already do that a lot. We've removed billions of tons of sugar and fat and salt from the food supply. And yet, as we saw earlier today from Professor Gibney, obesity rates are still high. So the question is, we can reformulate food. How do we reformulate human eating behaviour? That's sensory. And so I hope to show you that in the course of the next 20 minutes or so. So let's start by talking about odour, first of all. This is my colleague Sanna Bosveld, who's an associate professor in our team. Sanna's been doing work on smell for the last 10 or 15 years. This is just one study and an example of the kind of work we do. Here, what the interest was in terms of whether or not we could shape food choice behaviour by infusing an odour into a food choice environment and then see if it has an impact on the way people select certain types of foods. In this case, you can see that when a tomato odour is infused into the room, we see a higher selection of tomato soup, same for banana pie. And then indeed, there's bread rolls can be more selected if there's a bread odour selected or infused into a room. And we all have this experience if you walk past the bakery and you suddenly didn't know you wanted to have a fresh roll, and then suddenly you're in the shop buying a pack of fresh rolls. So we know this effect, and we've proved this many times scientifically with controlled studies. So odour cues can develop sensory-specific appetites and direct our choice behaviour towards those kinds of food sources. However, we've had a lot of hope about this area around 10 or 15 years ago, where we thought maybe it's also a non-nutritive or low-calorie way of stimulating the onset of satiation and reducing calorie intake. But odours don't seem to have a strong influence on the onset of satiation or indeed reducing calorie intake. It might just be useful for trying to redirect healthier choices. And it speaks to a differential role between, we'll say, visual and smell cues in terms of stimulating a food intake event versus, we'll say, texture and taste cues, which may be more linked to the onset of satiation or the onset of sensory-specific satiety. I could have many more examples of the odour work I could give you from today, but we want to move on to taste and talk a little bit about the work we've done in taste research and trying to understand how we map dietary patterns not in the context of nutrient intakes, but in the context of taste profiles. And this again was mentioned by Eileen this morning, not thinking about individual foods, but rather patterns of consumption and how we could target what is a healthy or an unhealthy pattern of consumption. This is my colleague, Associate Professor Monica Marsh. She's in our chair group, and Monica's specialisation has been in developing up taste databases. And to date, we've profiled the rated taste intensity of over 1,000 foods from the Netherlands, from Malaysia, and we've other food sources as well in the US. I should say we also have a separate related database on the eating speed of foods as well. So all of this is publicly available if you want to replicate any of this analysis yourself. The approach is based on standard traditional sensory approaches where we rate intensity with a highly trained panel. And then this gives us objective readings on the balance between different taste properties when you consume a specific food. And you might say, why do you do that, Ciarán? It's an awful lot of work. What can you use that for? So I'll give you an example of what we're calling sensory epidemiology. Sensory epidemiology doesn't think about the diet in the context of energy from fats or carbohydrates or proteins. We have all of that information already, and we have all those patterns. What we're more interested in understanding is how do diets taste, healthy or unhealthy, low or high energy? How do processed diets taste, minimally processed versus highly processed diets? What's the difference? Using this approach, we can insert these taste variables as dummy variables at the end of a nutrients data set, and then we can start to map dietary intakes. And this is just one example of work we completed in Singapore in collaboration with Rob van Dam in the School of Public Health. Here we were interested in the difference between low, medium, high, and very high energy intakes, and what was the relative distribution of energy consumed from different types of taste sources. So sweet, fatty, sweet, sour versus savory, fatty, different types of meals, or neutral foods, for example. And the working hypothesis at the time was that perhaps it was the presence of high energy intakes from sugar that was probably driving higher energy consumption. But we didn't see a huge increase in terms of energy from sweet foods. What we did see is a large increase in terms of meal size. Savory, fatty foods are actually driving the excesses in energy intakes. This is one example of where we can think about not nutrients, but rather foods and food choices, the factors that actually influence the way consumers select and consume their meals. We've also applied this subsequently to different types of population groups. We can look at children's diets in different types of regions. We can look across cultures. We've also done this for minimally processed and ultra-processed foods, if you coin that phrase, in terms of whether or not the taste-nutrient relationship is maintained across different levels of processing. And so it offers opportunities to think about taste in a completely different light. Another way we can think about using taste in terms of our understanding of nutrition patterns might be to think about how can we understand preferences around sweet taste. And this is the iSense randomized control trial that's been underway now for the past 4 years. It's just about to complete, or in fact it has completed, and the results will be revealed in October. The interest here was in asking, although we focus a lot on sugar reduction, the question still remains, should we be focusing on reducing the sugar content of our diet, or is it possible to retrain preferences for lower sweetness levels through exposure? Can you actually give people a lower sweet diet and they'll adapt their preferences to a lower level of sweetness? And so this was an incredibly comprehensive study where 180 participants were randomized to one of three arms, either the regular current sweetness diet, a high sweetness diet, or a low sweetness diet for 6 full months. So all of the sweet foods from their diet was provided for them. And then we have a 4-month follow-up. And the primary outcome is changes in sweet preferences over time. Can you actually change someone's sweet preference and bring it down by just reducing the sweetness of their diet? So I'm sorry to whet your appetite for this result. I can't give you the result today, but I'd be happy to share it with you in the future. So that's how odors might influence choice. That's how taste might influence your experience of eating and shape your diet. What about texture? And texture is an area I've been working on since I was here in the Nestlé Research Centre nearly 15 years ago. A lot of this work was pioneered by my predecessor in Wageningen, Professor Kees de Graaf, and also Monica Marsh. From there, they had about 9 PhD students working exclusively on the textures of liquid and semi-solid diets and how liquid and semi-solid foods in acute feeding trials. How can you play on those simple texture variables and do the influence intake? We saw incredibly consistent results. If you make food slightly slower to eat, you can reduce intake, and it has also a stronger effect on post-meal satiety. And it's already 10 years since we published our meta-analysis of this work with Eric Robinson, where we show that across 22 controlled observational feeding trials, and any trial where people ate at a slower rate, people tended to consume less energy. Interesting, because it means that people are eating in response to the foods they're being served, and they're eating slower, and not necessarily aware that they're eating less energy. And so I was offered the opportunity about 10 years ago to work in eating behavior in the Growing Up in Singapore to Healthier Outcomes birth cohort in Singapore. And there I led research on 4 to 6-year-old children, looking at eating behaviors around meal times, parental feeding practices, early life events, and how they linked to the energy intake acutely, but also to growth outcomes over time in Singapore. And it was, a truly fascinating time where I was able to access really impressive data from just children's ad libitum control trials. And we published a lot of papers, oftentimes in collaboration with our funder at the time, was also in collaboration with Nestlé Research and Development, Lisa Fries. And what we were able to demonstrate was that even in appetite-matched children, in this case nearly 400 children, appetite-matched children, those that chose to eat at a faster rate ate considerably more, sometimes up to 75% more energy within the same test meal for foods that are equally liked. And the children who ate quickly at one time point continue to eat quickly at subsequent time points. So it became a stable behavior that we could track over time. And it was linked to growth outcomes. So those children also had more rapid BMI growth rates and a more rapid adiposity growth. And we were able to tie this back to things like early life risk factors. We could tie it back to child inhibitory control or parental feeding practices, a range of factors around the early development of child eating behaviors that was linked to the emergence of this stable phenotype, which in turn predicted not just energy intake at a meal, but growth outcomes over time. So interesting, there may be a development of a behavior early in life that we can actually think about as a potential intervention target. And now with our current research in Wageningen, we've gone even further back, and we're interested now also in the development of oral processing behavior in the first 24 months of life, the emergence of deciduous teeth, and the physiological and anatomical changes that occur as a function of the textures that children experience in those first critical 24 months of life, because we believe that helps to set the kind of behaviors those children will exhibit subsequently. So we've shown acute trials that show eating rate drives intake, we've shown meta-analyses, we've shown cohort studies. What about at the level of the population? And this is again work we did in collaboration with the School of Public Health in Singapore, just where we profiled about 7,000 Singaporeans and just asked whether they eat fast or slow, and could see that indeed those reporting eating at a faster rate tended to consume more energy, tended to have higher body weight, higher adiposity, in fact a twofold higher risk of abdominal adiposity. So across a wide range of different cardiometabolic risk factor measures, we could see an association between reporting to eat at a faster rate and higher risk. And of course, we're not suggesting for a second that this is causal, but it does seem to be a very stable behavior that emerges, and across populations we've replicated this result in the Netherlands and elsewhere. We think this is something that's interesting to consider. And there's a lot of research in this area in Korea and Japan and around Asia. Eating at a slower rate is linked to much less risk. But there's a couple of ways you can eat faster. You may have a stronger drive to eat, or a metabolic or physiological drive to eat. But you can also eat faster by choosing foods that can be consumed at a more rapid rate. And we're interested in that interaction between the eater and the food environment that they choose to consume. And so, again, this is work from Singapore, where we looked at dietary energy intakes, but this time, instead of thinking about it in the context of taste, we looked at it in the context of the rate of consumption of that diet. And so here we've measured the eating rate of hundreds and hundreds of foods, and we have their energy density as well. And so we can impute this new value of energy intake rate, kilocalories per minute. And then using a wide range of different techniques, we can derive estimates of people's average energy from quartiles of foods with different energy intake rates, from, we'll say, low energy dense foods that are consumed slowly, right through to high energy dense foods that are consumed more rapidly. And using this approach, we can start to see if there's a significant difference across energy intake levels. So here we have a threefold differences in daily energy intakes of the 7,000 Singaporeans. But what we find is to get to the highest level of energy intake, it's not enough just to consume more energy density. Actually, it's energy dense foods that can be consumed more rapidly that were most likely linked to the higher energy intakes. Eating softly textured, energy dense foods makes it very easy to have a more passive consumption of energy within a meal, and this seems to be sustained over time because it's also linked to differences in body weight and BMI and a wide range of different metabolic markers. So the eater interacts with the food environment and the foods they choose. And I guess energy intake rate is an area that today in the modern food environment is under a lot of hot debate. And in the next section, we'll hear a lot about ultra-processed foods and the potential that maybe these foods are just too energy dense or easy to eat. And so one area that we were able to collaborate with was with Dr. Hall on his cell metabolism paper for ultra-processed foods. I can't believe this is the first time this slide has been shown today, because we've talked about it all morning, and we'll talk about it again later. But I met Dr. Hall before he ran his trial, and I asked him if he was considering measuring things like satiety, liking, sensory properties, and eating rate of the different diets. And he generously allowed me to include these measures in his randomized control trial. And for those that are not familiar, this is a 2-week or 4-week inpatient trial, 2 weeks on a minimally processed diet, and then 2 weeks on a diet that would be defined as ultra-processed if you subscribe to the NOVA classification. And the interesting result was a consistent change in energy intake of around 500 calories a day. This is a net difference, so it wasn't that people just ate more on the ultra-processed diet. They also ate less on the minimally processed diet. And I think it's interesting to consider that might have inflated the comparison somewhat. But at the time of the result came out, there was a lot of speculation as to why this difference was there and what was driving it. Is it something in the food? Is it something about the processing, the matrix? Is it some element that's disrupting a natural physiological response to the nutrients that are consumed? And actually, across more or less all of the nutrient, or all of the metabolic measures, there was really negligible differences after the different diets, suggesting that it might be something to do with behavior. Maybe it's the way people interact with these diets that might be driving that difference. And so again, we were able to look back at some of our own data, and there what we see when we look at our own measures from Dr. Hull's trial is within his subjects, when consuming a diet dominated by ultra-processed foods, they tended to eat those foods significantly faster. And because the non-beverage energy density of the diets was not correctly matched, or not fully matched, it led to a significant differences in energy intake rate, or kilocalories per minute. In fact, on average, about a 50% increase in kilocalories per minute when consuming an ultra-processed diet. Softly textured meals that are easy to consume, that are higher in energy density, might be potentially linked causally in the differences that have been observed in Dr. Hull's trial. And so we followed this ourselves with our own trials. We ran 2 subsequent randomized controlled trials just to test the hypothesis. Is it texture or is it processing that's driving the differences that have been observed? My former postdoc, Pezio Cho, what we did here was we developed 4 meals, 2 that were highly processed and 2 that were minimally processed. But we developed 2 that had faster, slower eating rates. So we had a mix of both eating rate and processing in our mix for an ad libitum trial. And what we could see was indeed is softly textured foods were eaten about 21% more in terms of grams and about 26% more in terms of energy compared to the harder textured foods that were consumed at a slower rate. Texture and the eating properties of the meals, not the degree of processing, is what drove the differences we've observed in intake. And this is a single crossover design for an ad libitum meal. My colleague Marlou Lauschlaut repeated this trial independently of us for a full day, looking at breakfast, lunch, dinner, and snacks, and again demonstrated a very similar result. The texture and eating properties of the foods, not the degree to which they were processed, is what was primarily driving those differences in intake. So something to think about in terms of it's not oftentimes front of mind for the nutrition community or for integrative physiologists trying to explain the modern food environment, but perhaps it might be the way we interact with foods as a function of their sensory properties might be driving the behaviors we exhibit. Now, of course, if you swallow that food and you eat it, you'll know that it changes also the matrix that's presenting as a substrate for metabolism subsequently. And one area we became interested in around 7 or 8 years ago with my colleague, Professor Marcus Stieger, was how do differences in chewing and structure influence the metabolic response to the consumed nutrient substrates? And so this is just a dynamic of what's happening whenever you put food in your mouth. I think you'd be familiar with this. We tend to grip food, we break it down, we push it to the back of our mouths between the molars for more efficient crushing. It creates mixing with saliva and initially fragments and then subsequently agglomerates in a sticky mass that can be swallowed safely. And this is well documented and well understood, and we have a parallel group that we work a lot with in the food oral processing domain where we try to understand a lot of the aspects around chewing and bolus formation and texture and structure. And it led to a strong interest in understanding, well, if chewing slower reduces your intake, what does it do to your bolus? And can it have an impact on glucose response or insulin response or even satiety or even your microbiome's activity? I don't have time today to get into all of the studies because we've done a lot of work in this area, but I wanted to make sure that I was aware that there's a lot of activity in this area in our group, that we know the texture at a macro level will influence the way you eat, your average bite size, your chews per bite, and the time food spends in your mouth. And we've been able to demonstrate time and time again that if we can slow that down, we can probably reduce your intake. But what about the bolus? We've shown actually that chewing for longer is influencing early glucose release, which in turn is having an incretin effect in stimulating insulin release. We know that on a calorie for calorie basis, a meal that is eaten slowly versus a meal that is eaten quickly one that is slowly eaten will have a stronger satiety effect. Now, these are not huge effects, but they are strong effects. They are reproducible effects in terms of feeling fuller on the same calories, and it's also linked to neuroendocrine signaling. Recently, we've shown that the size and shape of a food can also influence the bolus that's formed and its nutrient release. So we can see, for example, beta-carotene release and absorption can be influenced by the kind of shape and size of a carrot that you consume. And most recently, and I haven't got the slides today, I took it out because of time, but just to say we've shown that we can even influence colonic fermentation byproducts by changing the bolus surface area that's presented to the colon. So simply chewing the same mouthful of rice, a small bit versus a lot, can change the production of short-chain fatty acids in your small intestine. So interesting opportunities to think about Oftentimes when we eat, we don't even think about what's happening south of the mouth, but of course these are having potential knock-on effects. And we can't say at this stage whether these are clinically relevant, but they're nonetheless interesting to consider what could be the potential if we could align the eating behavior of the person and that specific food with the metabolic target we're interested in. Could we align structures that can actually optimize the metabolism profile we're looking for? I should just quickly highlight the picture I've included here is of bolus samples being collected and dried. This is a pretty disgusting area, so if you're thinking of getting into it, be aware you're going to be collecting a lot of sped out food and drying it in an oven. But fascinating to do, and if you're patient master students, it's great to do, but yeah, it's pretty gross. I wanted to include this slide. It's a bit of a paper I'm very proud of. If you think you understand something, well, the best way to test that is to try to explain it to a 10-year-old. So with my colleague, Mainu Korstens, who's in food process engineering, we've recently written a paper for Frontiers in Young Minds. I don't know if you're familiar with this journal. You write an article explaining a concept like where does food go when it goes in my body, and it's peer reviewed by 9 to 12-year-olds. So 9 to 12-year-olds give you comments on your paper, and some of the most intelligent comments I've ever had from manuscripts came from these 9 to 12-year-olds. But they asked us the question, from chew to poo, where does food go when you start to eat it? Where does it go in your stomach, and how does it go through your gut? And what's the impact of structure on that? And what's the impact of metabolism on that? And so we heard it earlier on today about protein digestibility. This is just another example of how oftentimes overlooked elements of the food matrix structure, the bread structure, or the plant-based protein structure, they can actually have a profound impact on what's available to the gut in terms of nutrient breakdown and absorption. And so we're interested in this idea of understanding that more formally. With Mainu, we've recently got funding to look at more advanced structures for fats and for carbohydrates, and trying to understand how we can engineer products that we can actually target the delivery of the nutrients to different parts of the alimentary canal for digestion, breakdown, and absorption, and see if it's possible to not just make foods that are tasty and appealing, but that also have a specific nutrient signature that we're most interested in. And with that, it's important also to highlight what was highlighted about 2 years ago when Remco Boom spoke here at the NINS meeting. He highlighted the huge potential we have in the future of using milder food processing techniques. We know that there's a very strong sustainability and even a cost reason to process our food more mildly in terms of dry fractionation or electrostatic separation. Instead of taking our raw materials and smashing them into 1,000 pieces, can we actually use milder processes that retain the innate structures of a food? And so with that in mind, about a year ago now, we started a new project called the MetaPro project, trying to understand the nutritional impact of milder processes. What's the impact on nutrient density of food, but also the metabolic profile of the food as it's consumed. And we do a lot of this work with plant-based foods, but also with carbohydrate sources, looking at the retention of innate structures in a food, or looking at intact cells that we can potentially add back to a food, and use that then to shape the kinetics of metabolism and shape the point at which glucose becomes available or not in the gut. So I've covered an awful lot, and I want to quickly summarize what I said. First of all, I think we often overlook sensory. We think it's just about likes and dislikes, but I hope I've been able to highlight that the sensory structures and properties, they influence not just what we eat, they also influence how we eat, and they even influence what we present to the gut for metabolism, so they can change the kinetics of our metabolic response to ingestive nutrients. And I think there's a strong future opportunity to align these matrix and texture properties with consumer behaviors and understand differences between people and target that by using structures that can optimize not just food intake, but also metabolic responses to those foods that are consumed. So I usually get, at this point in a talk, I'm about to finish, I have one last example, but I usually get the so what Ciarán question, which is all these acute trials and nice papers and cool graphics, they're all so what Ciarán. It's nice to think about that, but is any of this really applicable in the way we design foods or think about what people are eating and what they put in their mouth? Can you change behavior using sensory cues? We've actually been doing a number of proof of principle studies of this question for a number of years now, the first of which was about 12 or 13 years ago here at this very building in Nestlé Research. As a younger researcher, I worked in the metabolic kitchen and we developed meals. They don't look particularly appealing, but they were equally liked, and what we could see was that just by changing the format of those meals, we could reduce intake by around, on average, 12%. We repeated that trial many times since. Some of the examples are here. We've done at least a dozen of these acute feeding trials showing that if you change the physical properties of a food and the texture properties of that food, you can change the way people eat it, and you can change the amount they'll consume to fullness for equally liked meals and with equal satisfaction at the end of that meal. Simply by changing the way you eat it can change the amount you'll consume. And we went into a lot of detail. I haven't the time today to give you all of the background, but we've been doing a lot of modeling because testing meal by meal is a hugely time-consuming effort. What we want to really do is develop a formal understanding of what are the combinations of textures that might drive lower energy intakes. And so we profiled individual meal components and then built meals from those components and then predicted how quickly they'd be eaten, or even how much of them they would be eaten. And traditionally in sensory, we measure liking and we measure the sensory properties. We try to relate the two when you predict liking. We're trying to do something completely different here, because what we're trying to predict is how will consumers eat this food, how much of it would they eat in a free eating paradigm. And so we've been doing a lot of modeling in this area for many years now in terms of trying to predict texture-specific effects on eating rate, but also texture combinations that have the most effect. And in a recent paper we've just shown across 24 ad libitum meals, we're starting to get this right, I think, because we're now starting to be able to predict what we would expect to be a meal that will be eaten at a slower rate, and then this in turn is also linked to a reduction in average intake. And so about a 20% reduction in the eating speed of a test meal leads to a consistent 10% reduction in the intake of that meal. Not a huge amount in isolation, but it gives us an opportunity to start thinking about, well, how could I incorporate this into the design of healthier meals? If I combine this as well with energy density reductions or reformulation, we see this as an expansive effect. We know that slower eating rate with lower energy density has a very strong effect on overall energy consumed within a test meal. So we think of ourselves in sensory science then as the puppet master. We stand above the plate and we can actually move food around. We can actually move the way you will interact with it, change the average bite size you'll have, and change the amount you'll consume in total. We now have a reasonably good understanding of how specific textures can influence eating behaviours and intake. We understand the cumulative effect of that, and we start to understand now how to predict that behaviour. Just from an Excel file, we can start to predict, if we combine these things roughly, how much of it would be eaten a priori. So I think that's very exciting, and it's a new dimension for sensory in the future to start thinking about not just likes and dislikes, but how much of a meal will be consumed. Which brings me back to this question about ultra-processed foods, and we're going to have more of that in the following session, but I guess the Kevin Hall study gave us a lot to think about in terms of, is there something in this idea of energy intake rate or eating rate? Is it possibly moderating the effect? And even in the Hall study, is it the case that just this difference that's been observed, is it just purely an artifact of the differences in the eating speed of those two diets? We don't know. We have to test that formally. And so with that, we launched the Restructure project about 3 years ago. And this is a large-scale randomized controlled trial. These are our 3 PhD students who work on it. And here we're trying to understand the impact of texture and eating speed on the energy intake from ultra-processed diets. I won't go into the trial because it's nearly finished. It's ongoing at the moment. But it's a crossover trial with about 40 participants who are randomized to either a slower UPF diet or a faster UPF diet, followed by a 2-week washout and then randomized to the subsequent diet. Our primary outcome in this trial is simply energy intake from UPF diets. Is it different across those eating speeds? And if you believe truly in this idea that processing is somehow disrupting our relationship with food, well then in principle there should be no difference whatsoever in the intake of those two diets. But if there is a difference, we have to attribute it then to sensory properties of the meals, because everything else has been fully matched. And so our primary outcome is going to be energy intake, and we have a number of secondary outcomes and metabolic markers and exploratory outcomes that we're also interested in terms of glucose or metabolic flexibility and fasting hormones, body composition, etc. So I hope at a future NINS, someone from the Restructure team will be able to give you a formal overview of the overall findings, and the trial will be finished by the end of this year. With that, I have to conclude. I'd like to highlight, I think, that the dietary management of nutrition diseases rarely considers opportunities for sensory cues to moderate the flow of calories through our diets. I hope today I've shown you that texture, taste, aroma, they have a significant role to play in the way we select, consume, and even metabolize the nutrients we consume. These sensory cues don't just inform our choice behavior, they also inform our eating behaviors within meals, and over time can influence energy intake and our metabolic responses. And we had the question earlier in the panel about why it's so hard to move consumers to plant-based diets. It's important to remember that we can only really achieve the kind of dietary shifts we're aiming for in terms of healthy and sustainable diets if those diets are also appealing and satisfying to eat. And I think sensory cues are going to be the heart of that innovation in the future. So with that, I'm out of time, and thank you very much for your attention. I'll be happy to take any questions at the end.
To the questions at the end. Thank you, Dr. Forde. And our third and final speaker before we have the Q&A is Dr. Kosmas Dimitropoulos. Dr. Kosmas is presently at the Center for Research and Technology, Hellas in Greece. Kosmas is a principal researcher and really focuses on understanding a little bit more about a word we all talk about, and that is AI and GPT. He holds a diploma in electrical and computer engineering and a PhD degree in applied informatics. He has served for more than 5 years as the academic director of AI Move, which is artificial intelligence and movement for robotics and interactive systems. His main research interests include multi-dimensional data modeling and analysis, artificial intelligence, and human-computer interactions. Dr. Kosmas has been involved in several EU-funded research projects in the field of personalized nutrition, focusing on the development of AI-based nutrition recommendation systems. Today, he'll be speaking on using artificial intelligence to optimize meal plans to achieve dietary needs. With that, please give a warm welcome to Dr. Kosmas Dimitropoulos.
Thank you, Ryan, for the introduction. Thank you, Petra, and the whole group for the invitation. It's my pleasure to be here today to talk about the use of AI in the area of personalized nutrition. So I will talk about AI algorithms and how we design and develop AI algorithms in this area. Artificial intelligence is a hot topic. It has attracted a lot of attention the last couple of years. We all use AI algorithms, applications that use AI algorithms in our everyday lives for content personalization, for speech recognition, for image and video analysis, for search engines, and for so many other tasks. There are some terms that sometimes are used interchangeably, like artificial intelligence, machine learning, and deep learning. The term AI covers all the models and algorithms that enables machines to imitate human intelligence. So we have simple algorithms, which are based on very simple rules, to very complicated, complex mathematical models. Machine learning provides machines the ability to automatically learn. In this case, we need some data sets in order to train the mathematical models. Usually, we have to extract the meaningful information from these data sets, and these are called features. And in the machine learning, we call them handcrafted features. And then with this meaningful information, with these features, we train our algorithms. And deep learning is a subfield of machine learning based on the use of artificial neural networks, which consist of a large number of parameters, thousands, millions, billions, or even more. So in this case, we need large data sets in order to train these networks. In other words, in order to define the parameters of these networks. Now, in the area of personalized nutrition, AI is used for many tasks, for food image analysis. In this case, we use image processing algorithms in order to recognize the food in an image, or recognize the ingredients or the cooking instruction, or estimate the quantity of the food. Also, we use AI algorithms for analyzing the eating behavior of the user. In these cases, we analyze data from different sensors. In the simple case scenario, we have a video camera and we analyze the movements of the user in order to estimate the eating rate, which is associated with obesity and many other factors, as Ciarán explained in the previous speech. And also we have the AI nutrition recommender system, which provide nutritional advice to the users. In this case, this system analyzes the profile of the user. The profile of the user contains anthropometric characteristics and also information about the medical condition of the user, intolerances, allergies. And these systems provide personalized meal plans in order to cover the needs of the special user. There are different categories of recommender system in the area of personalized nutrition. In the first category, we have the knowledge-based recommender system. In this case, we design together with the nutritionist this system because we need to define the rules and in order to provide the personalized meal plans. In the second category, we have the data-driven recommenders. In this case, we use machine learning or deep learning algorithms, and we need to have a dataset which contains the profiles of the user associated with meals. And recently, we have the large language models like ChatGPT. These are foundation models trained on a huge amount of data. They can be used for different tasks, including personalized nutrition, and I will show you later on some tests, some experiments, and also some algorithms which use LLMs. Now, the first algorithm that I will present you today, it's a knowledge-based recommendation system, was developed within the framework of the Protein project, which was completed. It was a European project, a very ambitious project. Our goal was to develop an end-to-end ICT ecosystem that will provide nutrition support to users in their everyday life, at home, supermarket, and restaurant. The goal was to develop a mobile device, a mobile application connected with different sensing technologies, collect data and store the data in the cloud. Also, we had a dashboard for the nutritionists and the medical experts who could monitor the progress of the users and patients, and the goal was to provide dietary and physical activity programs. So we have all these ICT technologies in order to collect the data, and also we collected medical data from electronic health records, and also from some population we collected data from genetic, blood, and gut microbiome analysis. And the core part of this system was an ensemble of AI agents which run together in order to provide personalized meal plans to the users. Now, this is the AI-based recommendation engine. It consists of three parts. The core part is the ontology, because in the ontology we have model the knowledge of the nutritionists and medical experts. The input to the system is the profile of the user. So we have modeled the profile of the user. It contains physical characteristics, dietary choices, and different health conditions. And there is an inference mechanism. This inference mechanism exploits the knowledge that we have modeled in the ontology, and then rejects or promotes meals that exist in the expert-validated meal database that we created within the framework of this project, and this is a publicly available dataset containing meals from different countries. And the first part of the algorithm proposed appropriate meals for the user, and then at the second stage we had the daily plan generator, who used multiple criteria, such as the daily energy intake or the macronutrients it takes, the protein daily intake, and so on. And in this way, we generated the daily meal plan, a number of daily meal plans, and at the last stage, we constructed the weekly plans based on the preference of the user and the variability factors. I will give you more information about the ontology. The ontology consists of 6 different classes. So we have the class of food, nutrients, activities, conditions, medical condition, allergies, intolerances, deficiencies, different diets, and dietary well-being goals. And these elements of the ontology are associated each other with relations, for instance, relations between nutrients and foods. For instance, we have meat, which is rich in protein, or soya bean, which is rich in potassium, or rules about conditions, diets, goals in relation to foods, nutrients, and activities. For instance, type 2 diabetes has goal reduced sugar, or obesity has goal weight loss. So we had all this data, all this knowledge model in this ontology, and we used a mechanism in order to reject meals which are not appropriate for the profile of the user and promote meals which are appropriate for this specific profile. And then in order to generate the daily meal plan, we had quantitative nutritional rules, as you can see Here, for instance, for male, female, for adults with obesity, we had 10 different categories: healthy adults, adults with diabetes, adults with CVD, and so on. I will show you later on the populations that we supported. And at the last stage, as I said, we had two scores, two factors. One was the preference score. We collected data from the profile of the user and also from the history of the user, from the previous recommendation, if the user accepted or not the recommendation that we proposed, and the frequency score because we wanted to increase variability. We didn't want to propose again and again the same meals to the user. And in order to validate, because this is our goal, this was our goal to validate the system before we go to the intervention, we ran different experiments. First, we wanted to check if the meals that are inside the meal plans are safe for the user. So we created different profiles and we tested with different meals, and we see that the accuracy is 100%, so it rejects all the meals which are not appropriate for this specific profile, and sometimes we also reject more, but this is not a problem because our goal is to be sure that the proposed meals are safe. And then we created 3,000 user profiles, and you can see here the distribution of these users. We had healthy adults, healthy adolescents, healthy older adults, overweight adults, athletes, adults suffering from CVD, obesity, and diabetes, and with iron deficiency, and low fruits and vegetables. And the AI system generated 21,000 daily meal plans, and you can see here the distribution of the different types of meals. And the accuracy was pretty good, more than 92% accuracy in case of macronutrients and 85% for the other dietary components. And in terms of energy intake, again, the accuracy was more than 90%. Here you can see 92%, you can see the different population, and you can see the acceptable limits here with the red line. So more than 92% of the proposed meals were inside these limits. Regarding the variation, we had around 5 different meals per weekly plan, which was considered adequate for this test. Now, the problem with the odologies is that need a lot of time if you want to add a new meal, and a lot of effort, and also it takes time to process the profile. So if you have only one population without all these medical conditions, then you can use a simpler approach, and this is what we did in the Switch to Healthy project. This is an ongoing project which focuses on Mediterranean diet and aims to promote Mediterranean diet in different populations. There are 3 living labs in Spain, Turkey, and Morocco, and the goal is to develop some plant-based snacking products, some hands-on educational material, and also some digital tools. And this is our work to develop an AI-based application and also a digital game for the kids at school. Of course, the project focuses on families. I'm not going to talk about the families because it is a more complex problem. I will focus again today on personalized nutrition. Here, we redesigned the meal database. So instead of using the ontology, we created a new database with Spanish food meals, Turkish meals, and so on. But in this database, we included also the information about the allergies, about the dietary choices, the country seasonality, and so on. So we had a very simple selection mechanism. With one query, we could reject automatically the meals which are not appropriate for the user. And then again, we generated the daily plans. Here we have only healthy adults, so we exclude all the medical conditions. And for the weekly plan generator, because we want to promote Mediterranean diet, we had some rules regarding Mediterranean diet, so we wanted to reduce the processed meat, we wanted to propose more legumes, fish consumption, vegetables, and so on. And again, results with 2,000 user profile. You can see with Spanish user profile, you can see again that we have very good accuracy both in male user profile and female user profile, more than 98% in calories, and very good also accuracy in other macronutrients. Now, the main advantage of this category of recommendation engines, of knowledge-based recommendation engines, is trustworthiness and transparency. Because we develop together with nutritionists and medical experts these recommenders, it is easily to understand the outcome of the algorithms. The weakness is the high complexity, mainly, and also limited applicability because of the meal databases. Every time that we need to run an experiment in a different country, we need to design again a new meal database which will be suitable for the recommendation engine, and this is a lot of work. Now, recently we have the large language models, and especially ChatGPT, that can be used for providing nutritional information. It is very fast, and it has also access to an infinite pool of meals, and this is the main advantage. Many researchers have tried to evaluate the accuracy and the performance of ChatGPT in the area of nutrition, mainly in terms of nutritional content accuracy, in terms of the credibility of the generated meal plans. Many researchers have used ChatGPT for educational purposes, and others have tried to assess the accuracy of ChatGPT in the area of food appropriateness. In general, it is a convenient tool for providing nutritional guidance and education. It can also provide with some good accuracy meal plans, weekly meal plans, but again, there is a lack of knowledge on nutritional guidelines, and you will see that there are some problems. I will show you a study in a while. The main problem is with allergies, and most of the studies have found that proposes meals which are not appropriate for specific profiles. So this is a study that we did. We tried to compare ChatGPT, different versions of ChatGPT, with the knowledge-based recommendation system that we developed within the protein project. And you can see we created populations with obesity, CVD, and type 2 diabetes. You can see in terms of nutrients accuracy, the knowledge-based classifier is more accurate than the ChatGPT version. In the first experiment, we provided to ChatGPT the energy take of the profile, so we estimated the energy intake, the appropriate energy take, while in the second experiment we let ChatGPT to estimate it alone. And in general, ChatGPT works pretty well, but again, the accuracy of the knowledge-based recommendation engine is higher. And here you can see an example of a user. The green line shows, here we have the 7 days of the week, and here we have the energy take. You can see this is the green line represents the knowledge-based recommender, which is very close to what we say ground truth. You can see that ChatGPT sometimes has these fluctuations. That means that it doesn't propose every day meals with the same energy. And here you can see that the green recommendation engine is always inside the appropriate limits, which is not always the case for ChatGPT. So the idea is to use somehow ChatGPT because it is fast, and also it has access to many meals from different countries, from different cuisines, but somehow to align ChatGPT with the nutritional rules. This is what we tried to do by developing a generative model. This is a recent work. So what we did here was to use 3,000 profiles and 91,000 daily meal plans. So we created a generative model. The input here is the profile of the user. So we have a vector, and we have an encoder which maps this vector into a new space. This is called a latent space. And then we have a decoder which gets these embeddings and creates the daily plans sequentially. And in order to be sure that these daily plans are accurate, we took the nutritional rules and we created loss functions. So we trained the network with these loss functions in order to learn how to create meal plans which are appropriate for these 3,000 profiles. And we used ChatGPT in order to have access to different meals from different countries, and we asked from ChatGPT to give us equivalent meals in order to be able to support different cuisines. And then we generate again the weekly meal plan. It is difficult to interpret the model, but I like to show this geometrical space, this latent space, because you can see here that the profiles which belongs to the same population, for instance, adults with CVD, the green color, or adults with type 2 diabetes, here are together, are very close in this space. This means that the classifier, the network, has been trained correct. And in practice, what you see here are the mappings of these 3,000 profiles in the space. But in practice, the model has managed to model all this space, so there are no gaps. And that's why it is called generative model. If we have a new profile, an unseen profile, which wasn't part of the training set, the profile will be mapped somewhere here in this space, which now is in 2 dimensions. In practice, is ellipsoid of n dimensions. The new profile will be somewhere here, because we have modeled all the space. The decoder knows how to handle this profile and can create an appropriate meal plan. And you will see that we use this model with profiles from another study, and you will see that the system can generalize very well. The caloric difference is 0, so we had 100% accuracy, and regarding the macronutrients, the result was again very good, about 87%. And here we compare our work with ChatGPT. So initially we asked from ChatGPT to create weekly meal plans for Greek users, Portuguese users, Italian users, and you can see here the differences in calories and macronutrients, and then we use our approach with ChatGPT, and you can see that the difference in calories is 0, and that we have increased the accuracy of macronutrients. And here are some results with a study, with a real study, with real users from 2 studies, and we have healthy adults, adults with who are overweight, adults who are obese, and adults with CVD, and you can see again that we have very good results. This means that the algorithm can easily generalize with new data. And regarding the equivalent meals, we showed that ChatGPT can also create very good accuracy. Okay, the advantage of this approach is that we have access to different meals from different cuisine. It is very fast. We can easily integrate it, and the complexity is low. We can easily integrate it in different applications. Now, as a conclusion, okay, artificial intelligence opens up new opportunities for researchers in the field of personalized nutrition. Of course, we have not solved all the problems. There are many other challenges. The main problem is the problem of the multilingual food databases, but also we have to move one step forward, and we have to support sustainability, because until now we have focused on health, so we need also to support the other pillars, the other dimensions of sustainability, the environment, the affordability, and socioculture. So we need to insert this information in the network in order to propose also affordable meal plans and with low environmental footprint. Also, we have to go to precision nutrition. This means that we need to enrich the vector that represents the user profile by including information regarding the human gut microbiome, and also we need to enrich the meal database by including information about chemical components of the meals. And there are also ethical issues raised by the use of AI, because all this information that we collect is personal data, so there are privacy issues. And also there is a discussion about the sovereignty of the data. This is something that we study in another ongoing project, which is called Foodity project. And recently, the European Parliament voted a new legislation framework, which is called AI Act, and aims to classify the AI algorithms according to the risk they pose to the user in order to have safe use of the AI. We will see what will happen, and if other countries will also follow this example. And that's all from my side. Thank you for your attention.